involved in Lysogenic pathway of apoptosis because it is also component of electron transport chain and mitochondria.
Okay, so for this question, I just require you to remember the cytochrome in the intrinsic pathway is no more explanation for this. And the second, number 2. We want, please. Question number 2.
We choose cancer cells.
The cancer cells.
Because it cannot receive normal signal.
So, okay, for this question, I think the accuracy in both class is quite high because I think you understand my lectures, okay? So only cancer cells cannot killed by the apoptosis, because if we can kill them by apoptosis, there is no cancer occurs, right? BI12 or 362, please. Hoàng Quân.
Yes.
So please explain the question number 3.
I can't see the question for number 3.
Here.
Okay, so BCI2, it's kind of like a signal that could interfere in the apoptosis change reaction, and it will make the cells from undergoing apoptosis.
How this could block the apoptosis?
Okay, I didn't remember this part very well, but this particle can intercept in the apoptosis change.
So, okay, for this question, remember the BCL, this one kind of proteins, if you see the video clip about the apoptosis, you can see very well that 2 proteins, BCL, will block the BAX and BAKs. And because the BAX and BAKs has been blocked, they cannot go to the mitochondrial membranes and cannot change the permeabilities of the mitochondrial membranes. So therefore, the cytochrome C here cannot relieve from the mitochondria. So that's the important roles of the BCL2. So in my experiments, I also evaluate the levels of BCLs in my cells to see that apoptosis is really happened or not. Okay, so the next question, please.
Yes. Number 4. I'm sorry, but the screen, your screen is very blur. It's very. Name of the process which a myelinized cell spread throughout normal cells. Okay, so this process is called the invasiveness, because cancer cell from a tumor can spread through the blood vessels and invade other normal tissues. So it's called the invasiveness.
Okay, and so I will explain the 4 last question for you. Thank you, Hong Kong. So number 5, arrange the following sequences of cancer progress from a local tumor to widespread the metastasis. And for this question, so pay attention is answer in the fastest way. First, the metastasis is the final step. So in X answers, you find the ones, the metastasis is the final ones. This could be choose, or the other ways, mutation is the first step of the cancers formation. So if you find the number 2 in the first orders, it means this answer is choose. So 1 or either 1 in the last or 2 in the first. Okay, the right order of them is 2, 4, 3, 1s are mutations, or intervastations, tumor growth, and the last is metastasis. Okay, and the next questions: what is the following could be caused by the tumor suppression genes, or a protein that have prevent apoptosis? Knows tumor suppressor genes in the third step of them is they will produce or do activate the apoptosis. So there is no way that they produce the protein prevent the apoptosis. They really want apoptosis happens if we find out some abnormal cells, right? Protein can help prevent progression through the cell cycles. That's okay. A protein that form a path of growth factor signaling pathways, knows, and a protein that's called for DNA repair enzymes. This is not called for the DNA repair enzymes. So in the second step, the first step is they can prevent the cell cycles at the checkpoint. They stop the cell cycle at the checkpoint, that they prevent the progression through the cell cycle. So it's the first step. And the second step, they will stimulate the DNA repair enzyme, but they do not code for that. Because the genes is only produce the tumor suppressor proteins like P53, P21s, RBs. So it's not code for the DNA repair enzyme. These kinds of tumor suppressor proteins will increase and will stimulate the DNA repair enzymes. So for the next question, the embryonic stem cell can differentiate into which type of cells? Okay, so they can differentiate in the old type of cell because they are pluripotent or totipotent in the early state of this of the embryonic stem cell. Okay, a blastocyst I draw for you. That's when the stem go to meet the sperms. The sperm go to meet the eggs. We have the zygotes, and by severed cell division in the first step, they will produce a blastocyst in the very early state of the embryos. And inside the blastocyst, I draw a small region that's containing the embryonic stem cells. Remember in my previous lectures. And the last name or two defining the feature of the stem cells are two defining features. So the accuracy rate is quite low. You need to choose two correct answers. The first, the unspecialized cell when can specialize. So this is the abilities of the stem cells, and also cell renewal. Because besides or differentiate into another cells, they can also renew themselves. So at the end for the quiz today is a little bit hard for you, I think, but you need to get acquainted with them because it might appear in your final exams. To the lesson today is about one main cell type in the world that we forgot from the lesson number 1. This is the prokaryotic cells. Can you see my slide?
Yes, we can. Yes, we can.
Okay, so I received one comment that you want to see my face during my lecture, but I'm really sorry because when I give you a lectures, I use my laptop screen as a whiteboard. So when I give you lectures, so if I tell Osmond is not my face anymore. So the lesson today is about the prokaryotic cells. Is one of the main one of two main cell type in the world. From the lesson number 2 to lesson number 9, we only study about eukaryotic cells, and today's is a lesson about the prokaryotic cells. Also look at your cells deep inside your bodies, or you're not alone. It means that the bodies is some definition of the words, you actually consist both of the major cell types, prokaryotics and eukaryotics. And in general, human content no, it's not this. Human in general contains 10 power 14 cells, and 10% of this is a prokaryotics. It's just no, it's just only 10% of this is belong to yours. This means 10% is the human cells and 90% is the prokaryote cell, the bacterial cells. So you are home to 100 trillion bacterial cells. We are roughly 10 times more cells living in Earth than we do the cell make up our own bodies. And most of these outside cells are bacteria which living in your gut, in your gastrointestinal tracts, in your digestive system, and more than 100 species of them live in the colons. Its numbers of bacterial cells inhabiting in your bodies is 10 or 100 times more compared to your cells, and it occupies 1 to 3% of your body mass. This means that if you are 60 kilograms, you may contain 0.6 to 1.8 kilograms of bacteria, and the number of bacteria is 10 times higher compared to your body cells numbers. It's just because the cell size of the prokaryotic cell is quite small. Cells are in general, they are from 0.1 to 10 micrometers. So they have the similar size like mitochondria and protoplasts here also. So they come from 0.1 to 5 micrometers, whereas the eukaryotic cell from 10 to 100 is that's why we have large numbers of prokaryotic cells, but it occupies just only 3% of our body weight. And yes, the biggest cell in the world, the Archaea. And about the classifications of prokaryotes, prokaryotic cells, and we do have bacteria and Archaea bacteria. They are prokaryotics, and eukaryotic cells. So we have 2 cell types, pro here and eukaryotics here. Prokaryotes contain 2 domains, bacteria and Archaebacteria. So in the first lecture, in the first part of these lectures, I will give you information about the bacteria, and in the second part, I will give you information about the Archaea. So for the bacteria, they are really diversities in they're safe. First, they are most of them are in the root safe, spherical, or in the cup safe, and they may join together to form pair here. Pair the root pair root of the sphericals are they in the chains or cells in the packets of irregular aggregates. So they are really diversity in the safe, and also they in the habitats where they live in all the ecosystem in the world, in plant, animals, living or dead, or they live in the soil, water, in airs, in all the ecosystem, and also they live in the extremely environments, the hot or too hot or too cold, too salty, acidic, base, etc. And the extreme or environment the percentage of the bacteria is less than the Archaea. Remember, Archaea, they are extremely fine because they really like the extreme environment. So part of bacteria, they are live in the extreme environments, but it's not quite extreme like the Archaea. And they also the diversifies in the metabolisms. Based on the carbon source, we have the autotroph and also the heterotroph. The autotroph are known as the producers because they are able to make their own food from the raw materials, energies, like plants and algae. They are autotroph, and in bacteria we have autotroph also. And the heterotroph, as they are known as consumers because they consume producers or other consumer. And two different types of autotroph bacteria. We have the phototroph, where they utilize as the energy source, and the chemotroph, where they use the energy from the chemical reactions. We can divide the bacteria into two categories based on the oxygen required. We have the aerobics, means or they need oxygen to live, and another are types of bacteria, they are anaerobics. This means there's no oxygen days. And sometimes during the fermentation process, pay attention that your fermentation is requires oxygens or not. So like when you want to make the yogurt, this means they are aerobics. They don't require the oxygens. And based on the metabolized, based on the nitrogens or not, or they are sulfur containing compound, as we can classify the bacteria into subcategories. So we move to the main part of this lesson about the structures of the prokaryotics. They have four main components of eukaryotic cells, remember, plasma membranes, cytoplasms, genetic materials, the DNAs, and also the ribosomes. They share four key components to the eukaryote cell. But besides, they have the specific component that's only found in the prokaryotics, that the cell wall, the capstone, the plasmid. So here, cell walls out of the cell walls here. They have a cell walls, but there are different from the cell walls of the plants or the fungi. Remember, from plant they are cellulose, and from the fungi they made of chitin. But the cell wall, the prokaryotic cell, they are made from the peptidoglycans. And for the capstone, in another structure that covers outside of the cell walls. And also they have the extra DNAs we call the plasmid that have an important role in their life. And we have the fimbri is numerous. They are used for attachment of the hot cell and other surfaces. we have flagellum that is help to or bacteria move. They have some pillars allows bacterium to transfer the DNA molecule to another bacteria and also help them move. So we can go more details about the structures of the bacteria. About the structures, first about the DNAs or about the genetics materials. They are DNAs, but they are different. They are both double helix DNAs, DNA double strands. So sometimes we call them DNA double strand. But for the prokaryotes, they are naked. It means that they do not wrap around the protein histones, and they are in the form of circulars. While in eukaryotes, they are in the form of linear, and they are not like naked because they wrap around surrounding the proteins. Remember the proteins histones? We have an atomic core formed from two H2A, two H2B, two H3, and two H4, and they form the atomic core here. And also we have a linker protein H1. I remind you a little bit about the eukaryotics. In prokaryotes, they are naked and circular. That is different from them. It's really simple, right? Quite simple. And about the ribosomes, in the lecture number 3, I have already showed you the ribosomes of these prokaryotes and also the eukaryotes, right? For the prokaryotics, just remember for me this picture is a little bit details, but just remember for me for prokaryotes we have a 70S equal to 50S plus 30S. But for the eukaryotic cell, we have the 80S equal to 40S plus 60S. So just remember just only this. If I ask you the question about the structure of the ribosome, is only in this equation only. And for very special components of the bacteria, it is the cell wall. In the cell walls of the prokaryotes, they can classify into two groups, positive and in the negative bacteria based on their cell walls. First, about the positive bacteria, they have their structures. They have a cell membrane here, and they have a cell walls composed from the peptidoglycans. And the peptidoglycans is composed from the polysaccharides plus the amino acid chains. And we have another type of bacteria based on the cell wall we have. So we have a positive, this means that we have another type is negative. For the negative bacteria, they are the same with the positive. So we have the membranes, we have a peptidoglycans here, and then they have outer membrane. These peptidoglycans of the positives is very thick one, and this is very thin one. It's just, and these are even that they have a multi-layers, and these only monolayers with the negative, but they have the outer membranes composed of the lipopolysaccharide, and it is the lipids plus the polysaccharide, so they have a protects by one's more outer membrane. And when we stain these two types of bacteria by the staining grams, they resulting in the purples with the gram positive and pink or red with the gram negative because of this outer membrane. After the colorized by staining gram stainings, the outer membranes of the gram negative will keep the colors pink or red. So I have a table to compare between gram positive and gram negative. It's just based on their source. Gram positive is they have or is just single layer, just one layer of peptidoglycan cells, but it's thick because we have so many layers of the peptidoglycan, so it's very thick from 20 to 18 nanometer. But gram negative, they are double layered because they have the outer layers beside the peptidoglycans, but they're very thin. And here's thick layers of peptidoglycans, thin layer. Outer membranes, nose, here's presence, lipopolysaccharide absence and present. It's just because lipopolysaccharide is the structure of the outer membrane. It's just because the gram positive have no outer membrane, they have no outer membrane, so they more susceptible to the antibiotics. So if you have or you get disease caused by the gram positive, it means you have more chance to curve than the gram negative. So it's hard to kill the gram negative because they have the outer membrane. They have outer membranes. Okay. So when we're staining with the gram stainings, we have a purple with the positive and the pink color with negative. I give you also some examples of the bacteria gram positive like Staphylococcus, and 8 gram negative, Salmonella, and also the E. coli, very famous ones. Okay, they have also one structures. This is the components we can find also in eukaryotes, but in the next slides I will show you one structure that we can only find in the prokaryotes. This is the capstones. Here you can see the capstones is this one thick layers cover the cell walls. You can see them. They are the smooth surface and they have negative charge and they can prevent the phagocytosis, and we call them the virulent factors. If one kinds of the bacteria have the sticky capsule here, they will protect them against the desiccation, sự mất nước hoặc là làm khô. So because they contain waters and because they are sticky and help them to attach to surfaces in the environment, and they are smooth and prevent the phagocytosis, so it's really important to the bacteria. So think about this negative bacteria with the capsule, so it's really hard to kill them, right? And another structure that we can only find in bacteria is the plasmid. Plasmids, they are extra genetic material. So you can see here the main genetic material part. In the nucleolus, we have circular DNA here, right? We have circular DNA here, the main part. But beside the main part, in prokaryotes, they have the extra genetic material we call the plasmid. They still circular here, they have a circular, but they are much more small compared to the main genetic materials. And they can be transferred from bacteria to another bacteria. And when they using the pillars here, they can transfer the plasmid from one to another. And we also use the plasmids as a vector in the genetics engineering. We have a several types of the plasmids, like the R plasmids. They contain the genes responsible for to resistant to a drug. So our plasmids we call resistant plasmid. Like for example, these ones kinds of bacteria that contain our plasmid, they contain the genes and codes for the resistance of antibiotics, right? And the next factor plasmid we call the F. So the cells which possesses these plasmids we call the F plus. If they have the sex factor plasmid, we call the F plus. And the cell without sex factor plasmid we call the F minus, or they are recipient cells. F plus, they are the donor cell. And we have another type of plasmid. We have virulent plasmid. For the virulent plasmid, they are responsible for the production of virulent factors such as the adhesive factors, toxins, etc. We have also the heavy metal ion resistant plasmid. They contain the gene that resists to the heavy metal ion that may get from the environment. And I'll give you one example of the plasmid, how the bacteria can transfer these plasmids between one to another. Remember, we have a P-light here and we have a plasmid here. So the bacteria which have the gene want to transfer, we call the donor cells. And without the genes, we call the recipient cells and the F plus, F minus. So the P-lights, remember P-lights in the previous slides, they will form, they will contact to another cell and they will form a bridge here. And in the second step, one strand of the F plasmid transfer from donor cells to recipient cells. And the final step, they are synthesizing the complementary strains to restore the plasmid. So in this step, we have in refiners, we have two single strand, DNA single strands here. And in final step, they will synthesis to have the DNA double strand in X cell types. And recipients resistant cells and the donor cells synthesize a complementary strains. And in the final step, they both have the this kind of genes. So it's really important in the results of the antibiotic resistance. So you know that's for the antibiotic resistance cell, when we treatment the patient with antibiotics, there could be some kinds of mutation bacteria can survive. It means that they have mutation genes that can prevent our prevent the antibiotics effects, and they will transfer by conjugation. They will transfer the resistant genes into another bacteria, so they can generate one bacteria type or another generation that contain the resistant genes. This means that when people get that disease again, it's really hard to curb this kinds of disease by the old antibiotics. So we need to continue to keep our research to find out the new antibiotics, the next generation antibiotics. So it is plasmids is also very have the important roles in genetics engineering. I think in your high school you have already learned about these kinds of the recombinant DNAs. Remember that we use the plasmids from the bacteria to make a recombinant DNAs. So can you give me one example of this? We can produce big types of hormone that is really necessary for the patient.
We have insulin.
Insulin, yes. We have insulins and how they can produce the insulins. So for insulin they are required for the patient that have type 2 diabetes, right? Because they have an impairment and abnormal ways of the body's can regulate and yield the sugars, and now they require the insulin to keep the glucose in the bodies in the normal range, right? So before, in the past, they first isolate insulin from the duct pancreas and after that, in the industrial ways, we can extract the insulin from the capsules and also the pigs. So it was used for many years to treat the type 2 diabetes, and it could save millions of lives. But it wasn't perfect because first they are expensive, right? You need to receive the insulin from the capsule pancreas, from the pig pancreas, so we can take a small amount of insulins, but we need to perform experiment in numbers of the cattle and pigs. So first is expensive, and second, because they are insulins from animals, so it causes allergic reactions, right? We use the hormone from animals, so there is possibility that we can get allergies. So when they find out the function of the plasmids, we have another way to produce the insulin in the industrial. First, we have a plasmid from from bacteria, and in this way we use the E. coli, very famous bacteria. We take the plasmid from E. coli. We take also the insulin genes from the human. Insulin genes mean the genes encode for the insulin's storage information to produce insulin in the humans. So we take this part from humans. We cut the plasmid of the bacteria. They will open, and we will ligase. We will ligase the protein of the gene from the humans into the plasmid of the bacteria to make a recombinant DNA. So the blue part is the plasmid from bacteria. The orange here is the gene from the humans to make recombinant DNA tái tổ hợp. And by the next step, we will put back these plasmids into the bacteriums, but now they are containing the insulin gene from humans. And then they will multiply by several times, and we can culture these bacteria in the bioreactors, and we can receive high amounts of the insulin because now they have the ability to secrete the human insulin. So in this way, insulin is quite cheap and avoid the allergic reactions. So we can keep more patients, especially in the development countries like Vietnam. You know, insulin is quite high before, but now it's cheap and it's acceptable for the patients. So it's quite important to the mankind. Also, it's not only important to the bacteria. And so next, about the structures of the prokaryotes, we have a mesosome. Is a mesosome here. You can see that this membrane that's folding. Here is actually the membrane, just foldings. Actually, the membranes fold, and it is the extension of the plasma membranes into the cells. And if you have the formation of the cell walls, they have the DNA replications and distribution to the daughter cells. So when they replicate, they could stick in the mesosomes, and when they are synthesized in the two parts, the mesosome will make them go into the opposite poles, and That is the function of the mesosome. And our cells, like mesosomes, they are analogous to the cristae in the mitochondria. So in the mitochondria, the inner membrane just folds so many times to form the cristae. So it is the same with the mesosomes. So they also have the roles to help the cellular respiration and movement of the bacteria. They have the same functions like the cristae of the mitochondria. And for the next structures, it's not really a structure, it's like the formation of the bacteria in the harsh environments. When the environment is no water to live, no nutrition to live, some kinds of bacteria can form endospore, not about the spore about the. So in the starvation, when they get starvation condition, they will form the spore. And this process we call the sporulation, quá trình tạo bào tử, quá trình sinh bào tử. Here's how these ones bacteria can form the spore, can form the endospore. So the first, this is the our bacteria in the normal conditions. When they are hungry, when they have nothing to eat, when they get danger, they will form the spore. And this the first step. One septum we form septum vách ngăn. So one septum we formed near one post, and DNA replicates and extends into an osteofilament. In the step number 2, the septum separates the forespore. Forespore, the prespore is a prespore or prospore. Septum separate the forespore from mother cells, and DNA is pumped through the septum until reach compartment get the chromosomes. So new DNAs, mother DNAs. In the step 3, the mothers will engulf, eat these forespore, and by this and get is but no digestion occurs, right? We engulf, and now the forespore surroundings by the second membrane of the mother cell. And by the next step, the chromosome of the mother cells will be disintegrate. They would disappear. And the forespores develop a cortic layer of peptidoglycans between original spore membranes and the membrane formed from mother cells. COTS proteins deposited outer membranes. And in the next step, we have depicolinic acids which synthesize and calcium incorporated into the spore core. Depicolinic acids is the complex with the calcium, and they function to protect DNAs. Is protect DNA. Depicoline and calcium. It will protect DNA from the heat denaturations and the inserting itself between the nucleobase. Thereby, they can protect and also increase the stabilities of the DNA. So in the next step, mother cell will relieve the spore, and they will stay there, and they are waiting for a suitable environment to germinate, to become a normal bacteria. So in a general case, bacteria will keep dividing, and we have a multiplies of the bacteria. Do you know the several types of the bacteria can generate this kind of spores, like the bacillus and Clostridium, and mostly in the gram-positive. But I will show you an example that's familiar with. You know the Clostridium botulinum. Clostridium botulinum.
Có phải trong cái pate chay không?
Yes. Trong cái vụ pate chay minh chay đúng không? So it's because the Clostridium botulinum, they in normal case they are in the spore. They're in the form of the spores. Cells during the packagings or during the transformations or transportations or I don't know, is maybe there's problems with these kinds of parties, and these kinds of spore will germinate to become the Clostridium botulinums in these kinds of foods, and then they will produce the botulinums toxin, the most toxin in the world. So when the consumer consume that kinds of food, they will got the troubles. So that's a big problem. And another example. Do you know why when you want to sterilize your hands? So now we are in COVID pandemics, right? So when you want to sterilize your hands, we use the ethanol 70%, but not 90%. Do you know why? Or not actually 96 or 100%. Why we use 70%? 70 or 65 to 70s.
Well, because if the concentration is too high, it will it will destroy the membranes of the bacteria and it will prevent the ethanol infiltrating further into the bacteria.
Yes, this is one reason. And another reason, so why we attack them too fast with 100, so there is a possibilities of these kinds of bacteria we form the spore because it's too hot for them. And there is possibilities of these kinds of the disease or the pathogens, bacteria may form the spores. So we use the ethanol is more effective, but we do not use the 90 to 100%. So I will move to the next structures of the bacteria. Fimbres, pillars, and flagellums. I have a table that's compare the fimbres and the pillars here. The fimbres, you can see that there are numerous. Pillars are we also call the sex pillars is longer and thicker and is less numbers in the cells. And from the fimbriae, they are governed by the bacterial genes in the nucleus regions. Remember, the main nucleus is the main genetic material of bacteria. And the pillars, they governed by the plasmid genes. Remember the extra DNAs in the bacteria. And for the next characteristics, they are fimbriae. They are functions to attach to the surface attachments, and they enable the cell to adhere to the surface of the other bacteria. While the pillars here, they respond for bacterial conjugation, that they can transfer the DNA, transfer the plasmids from one bacteria to another. And for the fimbriae, no receptor to others, but the pillars, sometimes they serve as the receptor for the certain viruses. For flagellates, is just remember for me, they are have the movement of the bacteria only. And the next, I will introduce that the way bacteria can replicate. Or there's two ways for asexuals. For the asexuals, or we call the binary fissions, they will divide this. It's not really lies, but it's more like the same like the mitosis. That's from one cell. They are replicate the DNAs. Two kinds of DNA will touch to the mesosome here, and they go to the opposite poles. And the mother cell will increase the size, and then the cross will form, and we have two identical daughter cells. If we call the identical, is not really identical because the mutation rate of the bacteria is quite high. You know, all of these process happen in only 20 minutes. And 1 out of 200 bacteria is causing the mutation. So that's why when we treatment the bacteria with the antibiotics, there are high possibilities of them to create the new mutation strains, and they can prevent or they can go against our bacteria because the mutation rate in the bacteria is quite high. And this is the asexual binary fissions. We have another type of the replicates of the bacteria. For the sexual, we have three ways. First, about the transduction. There are some kinds of virus can attack the bacteria, and the transfer gene is may absorb or may transfer to the virus. And when these kinds of virus go to attack another bacteria, they will transfer again this gene from bacteria to another bacteria. We call that the transduction. And another conjugation is what I will introduce with you in the previous slides. And the last, we have a transformation. When some bacteria will kill, they will release the genes into the environment. And some kinds of bacteria can absorb this gene in the environment into their bodies, and we call the competent bacterium, and that's we call the transformation. And these are the three ways that bacteria can receive extra genetic material from environments, and we call them the sexual reproduction. So it's all about the structure of bacteria, how we can culture them. For the bacterial cultures, we can cultures on agar plates or in liquid cultures. And then we could studies and analyze this kind of bacteria. But up to now, 99% of bacteria in Archaea are uncultivable. Actually, we can say that less than 2% of bacteria can culture in the laboratory. And up to now, we have a total of more than 2,000 bacterial species has been cultured. Just only of them. For other part, they are still the mysteries because we cannot culture them. And most of the cultivable bacteria, if the bacteria inside of your bodies, in our digestive system, because we do know that's what they need for what there is nutrition, what is the pH values, what is the nutrition that they need, so we can cultivate them. For the other bacteria, we don't know, it's still uncultivable. For the genetic variation that I think I told you before, they reproduce every 20 minutes. 1 to 200 bacteria have mutations. So just remember this information is okay. And the next is the rows of prokaryotes. The rows actually here, row of bacteria. Because prokaryotes is containing the archaea also. The first is they are decomposers. They were recycling the nutrition from the dead to the living. And the second, they are the only organism on Earth can fix the nitrogen from the atmosphere, and this is important source of the nitrogen for it to use that in the root of the beans. The only organisms can fix the nitrogens. And also they have our digestions. We have numerous of E. coli in our digestive system, and they have to break down the food And also they produce vitamin K and vitamin B12 for us to use. So it's quite important. And also we can use them to produce foods like yogurts, right? Yogurts and medicines like insulin, right? And they are also pathogens. So besides the good thing, they are the bad thing also. Numbers of disease that's caused by the bacteria, even in the plant and also in animals, and numbers of disease that caused by our bodies, right? And it's just only 5% of the bacteria and pathogens. Right, is considered a good news. Okay. So how can we kill them? They cause the disease, so we can find a way to kill them. This is the antibiotics. Antibiotics is formed from the antibiosis, its meanings against life. Says a definition that the substances derived from the microorganisms are produced synthetically to kill or suppress the growth of other microorganisms. And the first antibiotics was discovered in the penicillins. Penicillins were discovered by Alexander Fleming. Accidentally, they discovered the antibiotics in 1928. So when he cultured the bacterium, he cultured the bacteriums, and we have a number of the bacteriums like this. But after he came back from his vacation, he found that numbers of green mold, penicillium notatans, that's grow here, and this inhibit or the killings kill the bacteria that his cultures. And they think that's okay. Penicillium notatans have benefits of killing the bacteria, so he can extract some kinds of compound from this kinds of green mold penicillin. I make noise here. I'm sorry. Okay, I mute him. So green molds we call Penicillium notatans is the first antibiotic that we for the human mankind. So it's curves so many trillion the persons. This is the antibiotics. So we can classify the antibiotics based on 3 categories. It's based on the mechanism of actions, beyond the spectrum of activities, and beyond the mode of action. So first, beyond the mechanism of actions, we can classify the antibiotics into several types. So one thing that inhibits the cell wall synthesis, we have a penicillin, cephalosporins, when they can inhibit the function of cellular membrane. If they can inhibit the function of cellular membranes, we have polymyxins. Inhibit the protein synthesis, we have chloramphenicons, we have a tetrathiones, and it inhibit the nucleic acid synthesis, we have quinolones, rifamycins, we have inhibit the folic acid synthesis, we have sulfonamides. So here I have the graph of the bacteria. We have antibiotics. Cells, just remember for me to nucleic acid synthesis. Remember for me quinolones 1, rifamycin 2, and for the protein synthesis, just remember for me chloramphenicons 4 and tetracyclines 5. Here. And for the cell wall synthesis, just remember for me penicillins 6. So they are, oh no, is it 3, 4? It's weird. So they are 5 categories. They are find antibiotics that I recommend you to remember based on the mechanism of actions. And for the next, based on the spectrums, we can classify into two groups: narrow spectrums and also broad spectrum. Cái này là phổ của kháng sinh ạ. Chúng ta có phổ hẹp và kháng sinh phổ rộng. Nếu các bạn xem TV nhiều hoặc là đọc báo nhiều hoặc là vào bệnh viện thì các bạn sẽ biết là kháng sinh phổ rộng và phổ hẹp như nào. Dựa vào phổ của kháng sinh thì mình sẽ chia thành phổ rộng và phổ hẹp. For the narrow spectrums, is it drug that only kill one kind or one stream of bacteria, like broad spectrum that they can wise antimicrobial scope. They can kill more than one strain of bacteria, and we have tetracyclines or chloramphenicol here. I have a graph to show you the spectrums of the antibiotics. We have penicillins, we have amoxicillins, we have the augmentins. So these two kinds of antibiotics, this familiar with your amoxicillins and augmentins. When you have a sore throat, the doctor will give you this one or this kinds of the antibiotics, right? When my son were 18 months, he had a sore throat. So it's the first time he needs to use the antibiotics. Even the first time, but doctor gave me the augmentins. So it's the broader spectrums, right? It's broader spectrums and they are stronger because beside amoxicillin, they have clavonate acid, so they are broader and they are stronger. But it's the first time my son is exposed to the antibiotics, so I asked the doctor to change into amoxicillins because they are narrow, they are not strong as the augmentins, but it's worked well is my son's case. And all the symptoms that's reduced and disappear in the first time he take this kinds of the antibiotics. So it's really important. So when you go to get doctors, when they give you the antibiotics, just make sure that your antibiotics is suitable to your conditions, so you can prevent the resistance of the antibiotic problem now. And also pay attention about the generations. Based on your condition, you should choose the generation that's the old generation instead of the new generations. It still coat yourself. Các bạn để ý có hai thứ mà khi các bạn dùng kháng sinh nhá. Thứ nhất là về phổ kháng sinh ấy, mình cố gắng dùng phổ hẹp hơn cho chính bản thân mình nếu mà nó vẫn có tác dụng với bản thân mình, đúng không? Như con trai cô 18 tháng bạn ấy mới phải dùng kháng sinh lần đầu tiên mà bác sĩ lại cho augmentin thì cô từ chối luôn. Và cô dùng luôn amoxicillin cho bạn và vẫn rất là ok, và bác sĩ cũng ok dùng việc đó. Vì ở Việt Nam có một cái là bác sĩ sẽ auto thường ấy, rất nhiều trường hợp bây giờ thì đỡ hơn rồi, nhưng mà ngày xưa là auto bác sĩ sẽ cho kháng sinh phổ rộng này, augmentin này, mạnh hơn này, hoặc là cho kháng sinh thế hệ mới, bởi vì cho một cái là khỏi luôn, và có một cái xu thế là người bệnh sẽ thần thánh hóa bác sĩ đó hơn. Thế nhưng mà bây giờ mình được học rồi, đúng không? Mình có kiến thức rồi, thì khi mà gặp bác sĩ ấy, thì mình cố gắng dùng phổ hẹp hơn và thế hệ cũ hơn nếu mà nó vẫn có thể chữa được cho bản thân mình, để tránh cái tình trạng kháng sinh trên toàn cầu nhé. So next, based on the here's the broad spectrums antimicrobials use may lead to the development of the super infections. Here is a normal case. We have normal microorganisms living with the pathogens, but when you use the broad spectrum for rộng, they can kill the non-resistant cells, but the resistant cells then can proliferate in very high rate, and it's called the super infections. The other partner is killed, so now these only resistant bacteria can grow, because now these only ones can use the nutrients and use the areas to grow. It's called the super infection. And for the next, based on the mode of actions, we can classify the antibiotic into two groups. Bactericidal, that's antibiotics will function to destroy or kill bacteria, and the bacteriostatics, and this will function to stop or slow the growth of bacteria, and then the immune cell will go to kill them, slow down the rate. It's only slow down the rate, and here kill the bacteria. And because you know that the bacteria and eukaryotic cells share some common characteristics. So when we use the antibiotics, we may have several side effects, right? And also because we have the good bacteria inside of our bodies, 10,000 times the numbers of our good bacteria inside our bodies, 10,000 times higher than the number of your cells. So when you use antibiotics, it will affect our good bacteria. So we have some side effect occurs. For example, the diarrhea, when you take the antibiotics, it will kill the E. coli or the good bacteria inside your stomach, inside your gastrointestinal, and it's leading to the diarrhea. So it says, right? Pay attention when you use antibiotics. Here I will give you videos about bacteria, and then we will have the 50-minute breaks, okay, right after these videos.
When I was a little kid, my parents would let me take my favorite set of dinosaurs into the tub, probably to persuade me into taking a bath. I remember my dinosaurs well. In fact, I used to know all of my dinosaurs. I had epic dinosaur battles. Sometimes my sister would play with them too, just a little differently. Anyway, I remember that one day, unfortunately, my favorite Stegosaurus lost his head. I mean, literally, it just popped off. And while I was determined to get my parents to fix it, we discovered that there was stuff growing in the toy. Now I realize what was actually large enough to be visible was likely mold, but at the time, my mother told me that Sergeant Stegosaurus would have to retire from the bathtub and I'd have to get a new one because bacteria had taken over. It led to two misconceptions I developed about bacteria. Number one, that bacteria were only found on or in things that had gotten contaminated somehow, and two, that bacteria are always bad, and that is why Sergeant Stegosaurus required replacing. Those are both not correct. First of all, there probably were bacteria in the dinosaur, along with the mold, but that shouldn't be surprising because bacteria are everywhere. Bacteria are found in our houses, yes, even very clean houses, and they are found outside. Bacteria colonize our skin, in our digestive system. That addresses the first misconception that you only find bacteria on contaminated or dirty surfaces. And as for the second misconception, well, bacteria sure do get a bad reputation. Now, I'm not disagreeing that the toy should have been thrown out. That was a good decision. There was probably mold that was in that toy dinosaur, a fungus, but there was likely plenty of bacteria growing there with it. A growing community of mold and bacteria, not ideal for a bath toy. But we do want to mention that many times all bacteria are lumped together as a bad thing, which shouldn't happen because not all bacteria are bad. In fact, many types of bacteria are helpful for organisms and ecosystems. We'll give you some examples. Some of the bacteria that colonize your skin are beneficial and actually help keep harmful strains and other types of pathogens from growing. Bacteria in your digestive system actually help break down food, and some can produce certain vitamins. Some types of bacteria are used in producing some foods that we eat. In ecosystems, bacteria have a very important role as decomposers. Bacteria also have major roles in the nitrogen cycle to fix nitrogen that plants need. These are just a few examples of helpful, beneficial bacteria. Now, that's not to say that bacteria can't be pathogens. Bacteria are the cause of strep throat, tetanus, tooth decay, some forms of pneumonia, diphtheria, salmonella, cholera. I could go on. Antibiotics can be used to combat some of these. And while antibiotics are very important for destroying bacterial infections, we shouldn't leave out saying that some broad-spectrum antibiotics can harm some of the good bacteria as well. Also, we should mention that antibiotics do not work on viruses. Viruses are pathogens that are not made of cells at all. You can check out our video on them. Additionally, there are vaccines which can prevent many types of bacterial and viral infections. Okay, so what are bacteria exactly? In the 3 domains of life, bacteria encompass one of them. They come in different types of shapes, as you can see here. Some of them are heterotrophs, meaning they consume or feed on some organic matter. But some are autotrophs. They can make their own food. Plants aren't the only autotrophs. Let's take a look at this bacterium here. Bacterium is just singular, while bacteria is plural. A bacterium is a prokaryotic cell, which are generally much smaller than eukaryotic cells, like our cells. If you recall from our video, that means bacteria do not have a nucleus or other membrane-bound organelles, but you will find ribosomes, cytoplasm, a cell membrane, and nearly all bacteria have a cell wall. Like all living organisms, bacteria have DNA. Bacterial DNA, while still double-stranded, is arranged in a circular shape. Depending on the species, bacteria can also have a flagellum to help with movement, a capsule which can help give them extra protection or attachment abilities, or pili which can help them with attaching to surfaces, including each other. Oh, and many bacterial species have a plasmid, which is basically like extra DNA. More about that later. Bacteria have some intriguing abilities that are different from our human cells that we'd like to mention. Unlike body cells, which perform mitosis and cytokinesis to divide, bacteria generally multiply even faster in a process called binary fission. Binary fission is a type of asexual reproduction, when the bacteria can easily divide to make a copy of themselves. Since it is asexual reproduction, the daughter cells would be expected to be identical to the parent cells, unless there's a mutation. Some types of bacteria do have the ability to share genetic material with each other. Remember how we mentioned that bacteria can have a plasmid, an extra copy of DNA with usually just a few genes on it? Well, bacteria can share these plasmids with each other in a process known as conjugation. The pili can be used to share this genetic information with each other. If the plasmid happens to have a gene that gives some degree of resistance to an antibiotic, this may allow the bacterium that received the plasmid to survive exposure to that antibiotic, which could be very problematic. You can learn more about how antibiotic resistance can develop over time in bacteria in our natural selection video. Bacteria can also pick up plasmids from their environment. Often when this happens, it's during a time of stress for the bacteria. In a process known as bacterial transformation, scientists can use a type of stimulus, such as a heat shock, to induce bacteria in a lab setting to pick up genetic material. There are all kinds of these uses for these genetically engineered bacteria that you can explore. Some types of bacteria can form endospores. Endospores allow bacteria to be survivors in all kinds of hostile environments. Lack of nutrients, freezing temperatures, drought, just some examples. This is a reason why hospitals have to be very good at sterilization processes. We won't go through the process of endosporulation or how bacteria reactivate after forming endospores, but this is definitely something interesting to explore. Finally, some types of bacteria, along with other types of prokaryotes called archaea, can be extremophiles. Unlike our own cells, extremophiles can live in extreme environments where there may be excessive heat, chemicals that our cells would find toxic, or even radiation. Overall, we share this planet with so many kinds of bacteria that scientists continue to learn more about them every day. And if this kind of topic interests you, you may want to look into the study of microbiology. So many careers, from agriculture to the medical field to environmental work, rest heavily on the study of microbiology. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Professor, we can't hear you.
We have 15-minute break now.
Okay.
Hi everyone, welcome back to the lessons. Can you hear me? Hello?
Yes.
Okay, so we move to the part 2 of the lesson today. Is about the Archaea. And the Archaeas, they are prokaryotes, right? Sorry. They are prokaryotes, and there's just one domain that is more sharing information in between bacteria and eukaryotes. Also, they still have no true nucleus, and they still simple, they smaller, so we classify them as the prokaryotes, but they have some characteristics belong to eukaryotic cells, and you can see that in that tree, the Archaeas is more close to eukaryotics compared to the bacteria. So I have a table to compare the characteristic between bacteria, archaea, and eukaryotic cells. First, about the cell walls, all the bacteria, actually most of, but we can call all the bacteria, have the cell walls made of peptidoglycans, whereas some of archaea have these. None of eukaryotic cells have the cell walls composed from the peptidoglycans, and another type of cell walls, another components to make the cell wall of the archaeas, we call them the pseudous peptidoglycans, and also they have the cell walls composed from the cellulose like plants. That's why we call them the share information between bacteria and eukaryotics. And the second characteristic is about the ribosomes. The ribosomes of bacteria, they are ribosomes of the prokaryotic cell, that is the 70S is composed from two subunits, 70s and 30S, right? And eukaryotics is, they have the ribosome of eukaryotes, like 80S, and is equal to 40 plus 60S. Archaeas, they sharing information in between bacteria and eukaryotes. Sometimes they are characteristic of the ribosome of the bacteria. Sometimes some strains have the characteristic of the ribosome of eukaryotics. And the third characteristic, the first amino acid in protein synthesis, as you learned in high schools, the translation process starts with the methionines, the first amino acid in the protein synthesis. However, in of the archaeas have the same characteristic in eukaryotes. Bacteria, they started with the methionines derivatives. The fourth characteristic, sensitivity to streptomycin and chloramphenicol, the sensitivity to some kinds of antibiotics. Bacteria, yes, but no for archaea and eukaryotic cells. And number 5, introns in the genes. So, introns is the part that brings no information. Exons have information, and in bacteria, very rare, or we can say there's no introns in their genetic materials. But for the archaea, yes, or some genes have the introns, and in eukaryotics present in many genes. And number 4 is about the histone presence. There is no histones, so they have naked DNAs. But for archaea and eukaryotic bacteria, yes, they do have the histones, histone atomic cores, 2H2A, 2H2B, 2H3, and 2H4 to make the atomic core of the histone. Yes, in archaea and eukaryotic cells. And number 7 is about the DNA polymerase. In bacteria, just only one kind, but in archaea and eukaryotic cells, several kinds of RNA polymerase. And this one characteristic that we call them unique for the archaeas, in the carbon linkage lipids in the cell membranes, in bacteria and eukaryotic cells, they link it by the ester. But for the archaeas, they link by the ether linkage. So we can call them unique for the archaeas because they link by the ether. here is a graph that's so used. Oh, sorry, I will increase. So here, this is the change the things. This is the membrane of the archaea. First characteristic, there is a linkage by the ether linkage, whereas with the prokaryotics and eukaryotics, they are ester linkage. And the second characteristic, that the side chains of the archaeas, they are based upon long isoprenoid chains with the multi-side branches. Here, they are multi-side branches. Whereas the tails of the prokaryotes and eukaryotes, we learned in the cell membrane lectures, right? And another characteristic is very specific for the archaea, that some kinds of the archaeas have the lipid bilayers replaced by the monolayer. They are monolayer, while in the bacteria and eukaryotics, they are bilayer. So in fact, they are fused two tails of the two phospholipid molecules to a single molecule. It's just actually they are two phospholipid monolayers in two phospholipid monolayers, one monolayer that have two polar. So they have two polar heads here, and this fusion make this membrane this more rigid and better able to resist the harsh environment. So that's why the archaeas can live in the extreme environments, and we call them the extremophile organisms, just because of very special membranes here, just monolayers, like fuse two phospholipid molecule into single molecule with two polar heads. So, and also the side chain, these number of branches here, another linkage. So they have the membrane that's very rigid, so they can live in harsh environments. So is all information about the archaea. Now you have a video about the archaeas, the video that's comparison between what difference between archaeas and bacteria, and also the function of archaeas in the ecosystem.
Have you ever imagined things with a movie trailer voice? You know, that dramatic voice that commands you to listen and be intrigued that shows up on some movie trailers? We think it could actually work great for biology. An example? In a world with no oxygen, one little yeast cell will do fermentation. Okay, tell me that does not make you want to learn at least a little about fermentation. If so, we have a video about that, and the yeast, which is a fungus, which can do it. But you know what would be the best topic for an exciting biology movie trailer? Archaea. Because they are probably some of the most exciting unicellular organisms out there, and many of them can live in extreme environments too. By extreme, I mean think harsh, but then more harsh. Areas with extreme temperatures or high salt levels. Though I should point out, archaea are not the only extremophiles. Bacteria can be extremophiles too. It's interesting because when I first started teaching, it was common to call archaea archaebacteria. But that is an outdated term because archaea aren't bacteria. Now, they do have some things in common with bacteria. They're both unicellular, they're both prokaryotes, so they have no nucleus, and they also don't have membrane-bound organelles. They both have genetic material and cytoplasm and ribosomes. Both bacteria and archaea can be classified as autotrophs or heterotrophs, and they both do binary fission to reproduce. Still, bacteria and archaea are two completely different domains in the three-domain system. That's a pretty big separation. What makes them different? In the three-domain system, you have the domain bacteria. the domain Archaea, and then the domain Eukarya, which has all the eukaryotes: plants, animals, fungi, protists. So let's look at the difference between bacteria and Archaea. Membrane structure is very different. Remember that all cells have a membrane. It's really important for homeostasis. If you take a look at our further reading suggestions, you can explore how the chemistry of the cell membrane of the domain Archaea is different. One example is that Archaea membranes typically contain ether linkages instead of ester linkages. You would find ester linkages in the membranes of bacteria and eukarya. Some Archaea have a lipid monolayer instead of a lipid bilayer. Turns out that a monolayer, as well as some other structural differences of Archaea membranes, can be useful for surviving in those extreme environments. You will also find a unique cell wall in Archaea. Recall that you will find cell walls in bacteria, and you'll also find them in Archaea too. Archaea cell walls can be very diverse, but they don't have this large polymer called peptidoglycan. You will find peptidoglycan in the cell walls of both gram-positive and gram-negative bacteria. While Archaea can also technically be gram-positive or gram-negative, most Archaea staining gram-negative, Archaea don't have the peptidoglycan. Archaea can have pseudo-peptidoglycan though, which is a little confusing, but it has a different chemistry to it. Like all organisms, Archaea have genetic material. Like bacteria, Archaea DNA is generally arranged in a circle. Although while bacteria tend to have one origin of replication, Archaea tend to have several. When studying the genetics of Archaea, it's interesting that now scientists find more similarities between the genetics of Eukarya and Archaea than they do Archaea and bacteria. So overall, these were just some comparisons of Archaea with bacteria. Keep in mind that most Archaea can't be cultured in the lab. That's because there is a lot unknown about what kind of media they need to grow on, or nutrients or environmental controls in order to grow them in the lab. Although that's a common challenge for quite a few types of bacteria too. Before we go, I want to mention my very favorite Archaea genus. So I have not been to a volcanic spring yet. Need to travel more. But Archaea members of the genus Sulfolobus have. They can handle temperatures that most organisms can't, like around 80 degrees Celsius, hot. And that's not all. This genus can also handle very acidic conditions. Substances such as hydrogen sulfide, which is dangerous to most living organisms, do not deter them. There's interest in studying how these Archaea could be used to treat industrial waste, and even convert parts of that industrial waste into something that could be useful. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
So we move to the part 3 that I will introduce supplementary information about the virus. It's for the virus is some ties in between living things and non-living things. So let's see what are these. The first, the histologies of the virus begins in the 18th century by Dmitry Ivanovsky. He discovered the first virus. Like at that time, we have a disease in the tobaccos. When he used, he green the leaf of the tobaccos affected by this kind of disease, and then they filtrate. They have a filtration. So they have a filtrate through this one filter that has a pore small enough to retain the bacteria. So bacteria will stay here, bacteria here. And he collect this kind of liquid to infect another another tobacco leaf, but is resulting in the disease. So he said that there's some pathogens, some things is smaller than bacteria, it can cause this kinds of disease, and because them is the virus. So this is the first time the virus were discovered, and they are the tobacco mosaic virus. We call them the TMVs. So one of the viruses, so you heard of, I think during these 2 years, because of the COVID-19 pandemic, so you heard a lot about the virus, and I think you have the servers, the first some basics information about the virus, right? So what are these? The first, they are the parasite, because they depend entirely upon another host, so they can't not live without a host cells, and they cannot be observed under the light microscope because they are too small. And they also have no internal cellular structures, because the structure of the virus is quite simple. It is much simple compared to the bacteria. They have no internal structures. They contain the genetic materials, either DNA or RNA, but never both of them. Is important information. And they have ability to replicate only in the living host cells, and they have no metabolism. So because no metabolism occurs, so they have no increase in the size. So they just contain DNA or RNAs and cut with the protein outside. No metabolism happens, no replications without the living host cells. They are parasite, and they are too small. That is all the characteristics of the virus. And the scientists now estimated that there are roughly 10 power 31 viruses. So in somehow, if you range up 10 power 31 of the virus and light them end to end, it looks like string like this. You have the columns that's with 10 200 light years into the space. So it's equal to 100 years. Now we call the light year into space. So you see that here's the amount of virus, right? And there's over 10 million times more viruses on Earth than the star in the entire universe. So you think that these kinds of viruses are waiting to infect Earth? Actually, it is not. Most of these viruses are found in the oceans, and they attack the bacteria and other microorganisms. It's not Earth. This is more amount of virus on Earth affected Earth, because they are huge amount of virus on Earth. Okay, so the next is show you some typical viruses. We have the HIVs, hepatitis B, we have the Ebola, we have the adenovirus, influenza, rabies, bacteriophage virus, rotavirus, herpes virus. So it's really, I think it's familiar with you, and this kind is quite famous virus, right? So you can see that the size of the virus is quite small compared to the bacteria, and compared to the eukaryotic cells, the size of virus is much more smaller. It's like it's more like smaller than ribosome. You can think that the ribosome is quite small inside the cells, right? But the virus is much smaller, so it's quite small. So why is small? Because they have very structures, or simple structures. It's just proteins caught outside genetics materials, RNA or DNA. So the protein codes or protein cells or the capsid, of the virus is made of. You can see here is the first part, is the first main part. They are made of many proteins molecules, and they will make a capsomers together to make up the capsid, and the capsid proteins are encoded by the genome of the virus. Not of the host cells, so that's provides or interaction for making them that's come from the genome of the virus, either DNAs or RNAs. And these kinds of virus we call the naked virus because they don't have the envelope here. The second type of virus, they are envelope. And their envelope is much more common in the animal viruses than the plants. And just because these kinds of the envelope, they are lipid bilayers, and the coverings are the virus, and we call them the envelope. And they are derived from the nucleus or cytoplasmic membranes of the previous host cells. And pay attention, we have some kinds of spines protein here. You know that the coronavirus, they are envelope virus. They have another envelope surrounding, that's two are here. There's two main structures of one virus here. And this is the three optional structures. And coronavirus have this kind of structure, and also the spike proteins here. And this envelope is come from previous host cells. And the envelope is more susceptible than the capsid to the environment pressure. And the virus needs to remain the moisture in order to survive. So a virus can recognize the cells and infect by matching the surface and receptor on the cells, and they can only attack the certain type of cells, and they are said to be specific. Like you have a surface here, you have some receptor here, and if the virus can match the receptors, they can invade this kind of the cell type. So here's if the suckers can only attack these receptor, have the shape of a square, can only attack the cell with these receptor surface like squares, circles, and triangles is only attack the receptor that have the shape of the triangles. So an example, that's the only rabies rabies virus can attack the brains or nervous cells, but no other cells. And the coronavirus can only attack the lung cells, right? And next, how they are very simple and how they can replicate. First, they can only replicate inside a living host cell. Living host cell. And there is two pathway. The first is the Lytic Pathway, and the second is the Lysogenic Pathway. I think yours are already learned it in your high schools. Yes. So this is the pathway that they draw it in the phase or virus or bacterial phase. So this is the bacteria. The virus here attack the cell membranes and then relieve the genetic material, either DNA or RNAs, into the cytoplasm of the host cells. And then there's two pathway here. One in the Lytic pathway, they can replicate the DNAs and also synthesis the new particle, the new proteins inside the host cells, and they can packaging into the new virus, and they can relieve themselves into the environments by break down the membranes of the host cells. And the second pathway is lysogenic pathway. Tiềm tan, chương trình tiềm tan. So when the DNAs of the virus can integrate it into the DNAs of the host cell, and then the host cell that bring the DNA of the virus that's multiplied, that can divide. So we have the new generations of the cell that containing also the genetics material of the virus. So when the cell gets the stress or some other effect from the environment, they get stressed, and the genes from the virus will activate, and they start back to the Lytic pathway. So here is this complete process of the Lytic pathway. One is the virus attachment the host cells, injection the DNAs, penetrations by early protein synthesis. Here, the host cell DNA is degraded. The DNAs of the virus replicates. They synthesize also the proteins. They assemblize to make a new virus, and then they release. I introduce here the two types of the virus that you need to pay attention in the no, I always talk about the exam, sorry. So two types. First, bacterial phase. That's the virus only infect the bacteria. That's you know, that's T for bacterial phase. Like in the high school, you have already learned about this. The TMVs, causing the TMVs, right? And the second is the retrovirus. We have the risk presenter, like the HIVs. So for the HIV, the first, they will bindings in the surface of our CD4 immune cells, and then they can relieve the RNAs into our cytoplasms. Because they are RNA, they need to reverse transcripts into the cDNAs, and the cDNAs will synthesize complementary strains into the double strained DNAs, and then these kinds of DNA will integrate into our DNAs cells or in lysogenic pathway, right? So in some case, these DNAs containing the gene from the virus can produce the mRNA of the virus, and also the mRNA of the virus to produce the proteins of the virus, and also the RNAs of the virus. And then they will packaging and binding and become a maturation virus, and to attack another CD4 cell types. So pay attention, we have a retrovirus. This means they containing RNAs. They need to transcribe into DNAs. So we call them retrovirus. It is a virus, but we have the other thing that is not virus. It's just the proteins, but they can cause a disease like the virus, and we call them the prions. And the prions is the proteinaceous. They can infect infectious particles, and they can cell replicate, respond for a range of neuron degradative disorder in humans and mammals, and they causing the DSA, the mad cow disease, DNA. So the prions, they always affect the brain, and is always causing the death in the cows. So I have a question, that's how could something that's contain no nucleic acids can be capable of replicate itself? So now they are proteins. That's how this kind of protein can replicate. To causing this disease.
I think it can be an enzyme that catalyzes another normal protein to become an abnormal prion.
An abnormal what?
An abnormal protein. It can be an enzyme or.
They are enzyme. They are enzymes.
I think
Before they are enzyme.
no, they are not in. They may not enzyme, but they can catalyze the product, the process that turn a normal protein into an abnormal protein.
So this is one. Actually, we cannot find the mechanisms. That's why that's one protein that can't replicate. And this case, you're right. They are not replicated at all. They are appears as the alternative versions of the normal animal proteins, and somehow they have ability to cause the normal version to refold itself into the mutation form. So like a series of reactions, they can make a normal version refold into the mutants, and that's why the prions can propagate itself. And another type of is not really the virus. A part of the virus can cause a disease. We call them the viroids. A viroid is normally they are plant pathogens, that they are only single-strand RNAs, and they do not code for any protein products, but they can cause the disease in plants. So we call completed virus. We call them the virions. And we have also the single-strand RNA cause disease. In the plants, we have the viroid. And one protein that can cause the disease, we have the prions. That is three forms that can cause the disease. One completed virus, one is RNA single strands, or one is proteins can cause the disease. So we have here a table that's comparing the virus with the living cells. About the structures, the virus have the genetic material that's either RNA or DNA, but never both of them, and they have a protein coat. And of the living cells, they have the four common components: the membrane, cytoplasm, genetic materials, and other organelles. And in the prokaryotes and eukaryotic cell, they share one organelle, that is the ribosomes. For the reproduction, they copy itself only in living host cells. But for the living cell, they can multiply, they can replicate by themselves, either with the asexual or the sexual ways. And for the genetic materials, DNA or RNAs, never both of them. But the living cell, we have a DNAs as the genetic materials, and they can we have the transcription process to produce the RNAs. For the growing development, no. And in living cell, yes. So because they have a nose or metabolism, so we have no obtain new energies from the external environment. They don't need to do that because they all use the everything inside a living host cells. And the living cell, yes, they need to obtain new energies either from outside, from the light, or from the chemical reactions, or they can use from other composers. Respond to environments, no. But the living cell, yes, you can send the environment is hot, cold, it's windy, or it's cloudy, spring, etc. And because there's no metabolism, so the virus is no change over time. So it's mean change in the size and the shape, but yeah, they have the mutations also, so they can change by the mutation. But here is more mentioned about the growth. It's not about the genetic materials. And for the living cell, yes. And here's how many characteristics of life do the viruses possess. Maybe you can think it's at home. And we have another question, that's, are these kinds of virus living? So it's still debatable. That is, one side is called them the living cells. One side is they are non-living because they cannot replicate, they have no chain, they have no metabolism system. And me, in between 50-50. So they are in between living and non-living things. So ở sách giáo khoa tiếng Việt mình có là nó là dạng sống á, không được gọi là sinh vật sống, nó chỉ là một dạng sống. And what is the roles of the virus in our ecosystem? So first, they are harmful because they cause disease. They are the pathogens, and we have numbers of disease caused by the virus. Like we have a common colds, we have flu, smallpox, Ebola, herpes, AIDS, chickenpox, rabies, so many human disease. We can prevent the virus by the vaccines, but are not treated with the antibiotic. Antibiotics is only treated the bacteria. And so you guys come from the ICs and also the cybersecurities. Can you tell me the common between the virus in the IT fields? What is common between the virus which affects our laptop and the virus which can attack our bodies? What is common between them? So you guys come from the ICT and cybersecurities. What do you think that's the common point between the virus attack our bodies and the virus attack our computer? Yes, Bùi Hoàng Huy, please.
I think the virus from the computer also harm to the computer, and the virus from human can also harmful to the human.
Do you think the virus in your computer have any beneficial?
Yes,
We can use.
We can use virus to create more prevention.
Okay, so this is the virus in computers. So what are the characteristics that you think that they share the same characteristic?
I think they are
For the harmful, but I think they are beneficial also, right?
Yes, they can be used to making your system more security.
So, what happens when they invade your computer? And what happens when the virus invades our bodies? When they attack our bodies, what happens?
They will both be like prevented by the anti-system of the computer.
So for me, I think they share the characteristic that they are the commander, right? The commander, they are reprogramming ourselves. Reprogramming ourselves to become their factory to generate more viruses. You think that's these kinds of characteristics with the virus from computer? Do they do the same? They use our computer to generate more virus and to invade other kinds of the computer. It is the same characteristic between them.
Yes.
And also, beside the harmful, they share the best beneficial that we can use, like in genetics engineering. The harmless virus can carry the good gene into cells. You know that the AstraZeneca vaccines. For AstraZeneca vaccines, we use the harmless virus to carry the things or the genes of these coronavirus to insert in our bodies. So that's what we use the harmless virus. And another type of genetic engineering that's in the gene therapies, we can use the virus to bring genes we want to insert into the genes of the human cells. So many beneficial of them beside the harmful. We have also a video of the virus, but maybe you can watch it at home. I will show you here also is about the AstraZeneca and is the way that we can produce the mRNA vaccines, like the Pfizer and also Moderna. This is the ways we can produce the vaccine to prevent the coronavirus. The first is the mRNA here. Do you know what this kind of mRNA encode for? mRNA here is actually mRNA of the virus, and this can synthesize the spike proteins of the virus. And also, this will be enclosed by the lipid monolayer here. Do you know what's LNP here? What is the LNP here? What's different from the lipids or from our bodies? So the student come from the nano programs, nano department. They are lipid nanoparticle. And what the scientists in the nano fields can develop. So is your work in the future, is one sign of your work in the future. You can develop and you can discover some kinds of this. And these lipid nanoparticles can cover these kinds of mRNAs. So okay, we can cover and bring the mRNAs here to, and then we have the vaccines. When we insert this kinds of vaccine inside of their bodies, these lipid nanoparticles will be engulfed by our immune system. And then after they will relieve the mRNA of the virus here, and these kinds of mRNAs can translate into the protein of the virus. So you know that these kinds of proteins is antigens. They are strains, right? And then ours are immune cells, macrophage cells, will send them into MHC receptor. And they will connect with another T cells by no distance. Remember about the cell communication lectures? I show you this, right? We send the antigens into the MHC receptors of the macrophage. The T cell will come and receive information by no distance. And then the T cell will activate the T killer and also the B cells to produce the antibodies. Kháng thể. And also B memory. So next time so antibody will protect our body from the coronavirus from the 3 to 6 or 12 months is not defined yet. And the next time when we have been attacked by the coronavirus, the B memory cell will produce more antibodies to protect our bodies. So that's the ways that humans can produce some kinds of vaccine to prevent the disease from the virus. So it's the end of the lecture today. If you have any questions, you can ask me in the tutorial class. This is the final lesson that we have. We have another lesson about the cell cultures and replications in next week. Today we have no attendance checks like a present to all the students because we have the 11th lecture. So I just slow down into 10 attendance checks only. Today we have no attendance checks. So you have any questions? If not, we will stop here. So see you next week. Goodbye.
Cô ơi, cho em hỏi là có em là phải đi quân sự, em ở trong khóa trước phải đi quân sự ấy. Thì
cái đó có bạn nào hỏi và cô đã email rồi ý. Thì là buổi sau thì cái phần recording của cái bài sau, thực ra bài sau nó rất là ngắn thôi. Các bài này là 3 tiết nhưng mà bài sau chỉ có 2 tiết thôi, và cô sẽ record và cô sẽ up lên Moodle cho các bạn đi quân sự. Có thể access vào xem.
Hôm nay có bị tính là no attendance không?
Không, nếu mà có lý do như thế làm sao mà tính là đỡ attending. Nha, đối với các bạn đi quân sự thì có thể ok, so với bài sau không vấn đề gì cả.
Có phải gửi mail xin phép không ạ? Hay là như nào?
No, không cần đâu. Cô có list của các bạn rồi. Cứ bạn nào BA 11 là phải đi quân sự rồi. Cô sẽ có list riêng của các bạn nhé.
Ôi mà em là bi.
Vì tôi cũng không biết, cô sẽ có list của các bạn và phòng GD sẽ gửi cho cô, không vấn đề gì cả. Đối với tất cả các bạn đi quân sự rất là không phải lo về vấn đề quân sự nhé. Nhà trường đã cho các bạn đi là chắc chắn là sẽ có giải pháp cho các môn học của các bạn rồi. So, alright, goodbye for now and see you next week.
Can you give me
Goodbye, teacher.
the form to
Goodbye, teacher.
attendance form?
Form for what?